difficult topic very clear to many of us. Thank you very much. We're going to move on now to some comments from patient groups and our industry partners. But we're going to start with Muriel Finkel. Is she here? Hi, Muriel. Welcome. Where are we putting her? Oh, I'm sorry. Thank you, Matt. This is a good spot. Welcome to Chicago. Thank you for coming and being with us today. We appreciate having you here.
Thank you. I traveled all the way from Wooddale, Illinois. It was a trek in rush hour, though.
It is.
Hi. I'm Muriel Finkel, and I'm co-founder and president of Amyloidosis support groups. We run face-to-face support meetings in more than 25 cities for all types of amyloidosis. We've met more than 5,000 amyloidosis patients and caregivers since our first meeting in 2004. Some attended our meetings in person, some call our hot helpline, and some take part in one of our many social media groups. In our letter to ICER, we shared the experiences of several hereditary hATTR families we met at a few of our support meetings in the early 2000s. Some had minor symptoms at that time, and some were pre-symptomatic, but all had watched parents and loved ones suffer and die from the disease. There were no clinical trials available or even being talked about when we met most of them, and liver and/or heart transplants were the talked about treatments. All the patients we highlighted in the ICER letter were dead within 10 years of our meeting them. They were not old. They were in their 20s, 30s, 40s, and 50s. Some had organ transplants. One woman had a heart, liver, and kidney transplant and made it about 5 years. Some went from walking with a cane to a walker to a wheelchair, from diarrhea to diapers to agoraphobia. Leaving the house became an overwhelming chore for most of these patients. Most were dependent on others to drive them to meetings as well as doctor visits and the grocery store. They shared that they never knew when the diaper would just not be enough. We saw this become a burden to their minds as well as their bodies. Their caregivers were also deeply affected by these symptoms. Everyday chores became almost impossible, and many of the caregivers shared with us their frustrations. Some even walked away. Our patients left behind families including children, siblings, nieces, and nephews, many of them also vulnerable to hereditary amyloidosis. Imagine the guilt a parent has knowing they may have passed on this insidious disease to a child. People talk about cardiomyopathy as if it is the worst thing that can happen with hereditary amyloidosis, but we have seen neuropathy impact quality of life just as much. And speaking of cardiomyopathy, why does everyone try to make these two separate diseases? FAP, FAC, it is one disease, hereditary amyloidosis. Most of our patients have both organ systems involved. If they don't, it appears they soon will. Often the African American variant, ILE 122, is referred to as a strictly cardiomyopathy variant. We have seen families destroyed with this variant, and several have attended in wheelchairs due to neuropathy. Many of our ATTR patients present with carpal tunnel syndrome several years prior to diagnosis. We've been told this is related to neuropathy as well. Now let's fast forward to 2018. We now have had patients take part in successful clinical trials for both patisiran and inotersen. As an observer, I now see patients living with this disease rather than suffering and dying with it. These patients tell me that they are concerned that the treatment will no longer be available due to possible insurance and/or financial issues. I read recently that our armed services are made up of close to 50% racial minorities. Many of these young people will be dependent on the VA for their medical services for the rest of their lives. We've got to make all these new treatments available to them through the VA and TRICARE. We cannot forget the poor and disenfranchised that have no hope other than Medicaid. We have teachers with retirement plans with smaller insurance carriers. There are many that are opt for Medicare Advantage or an HMO plan, such as Kaiser, Blue Cross Blue Shield's HMO, Anthem, Aetna, United Health Group, Cigna, Humana, WellCare, and more, because it is less expensive. We've got to make sure that all these plans and others cover these treatments. We've been told that too many might be stuck with a 20% copay. That can be as much as $9,000 per year. What good is having a treatment if only the wealthy or non-military government employees have access? Our hope is that these treatments are available to all. I'm available if any of you have any questions.
Muriel, just as a point of, thank you very much. As a point of procedure, we ask folks if they have any conflicts of interest to declare as when they make their comments. Do you have any conflicts of financial or otherwise regarding this particular topic?
I did report that on paper. I'm 72, so the memory's kind of shot. But
I got it. Check. I did not turn around.
Amyloidosis support. Oh, okay, you've got it
right
up
there.
there.
I got it. Check. Done.
Oh, good.
Nothing in
okay.
addition to that.
So you've got it all there. Thank you.
All right. Thank you.
Appreciate that.
Thank you, Muriel. All right. Our next presenter will be Charles.
Any questions, by the way?
Sorry.
No questions, no comments? Is that a good thing or a bad thing?
That's the way it usually works. It's good. It's very good.
Okay, thank you.
Thank you. Our next presenter is Horwath. Charles. Thank you, Charles. Thanks for coming to Chicago.
First off, for as big as this thing is, 5 minutes doesn't seem long enough, especially for the way it seems like it's going here. But anyway, I'll start with thanking you for the opportunity today. I see it's very important, they said. It is very important. It says, I have a lot to tell you in just 5 minutes, so quickly let me say this disease has placed a very big burden on me and my family, all mentally, physically, and financially. Plus, this has been a difficult disease to diagnose. My family owes my Uncle Bill for time to get diagnosed and to learn his condition. Otherwise, we wouldn't have known as much as we know today. Bill went to doctors from Seattle to Los Angeles until someone finally suggested to go to the Mayo Clinic. He did, and he finally got a proper diagnosis from Dr. Maury Gertz in the early 1990s. The condition manifested in my Uncle Bill. It also appeared in his brothers Jack and Fred. When my mother started showing symptoms, and they were getting pretty bad, my wife Cheryl and I moved her in with us. The last months of her life, maybe like the last 6 months of her life, we had to bathe her in bed, change her diapers, medicate her, and try to get her to eat. She was in so much pain, and she was so sick, it was really awful for all of us. She asked me many times if I could take her life. I apologized and said it's just something I can't do. Anyway, she passed away in my home as she had wished, weighing about 82 pounds, and she was a pretty good-sized gal. As I turned 60, I couldn't deny or hide it any longer. I was starting the same symptoms my mother had. Nausea, vomiting, and diarrhea were already pretty bad. Over the next 3 years, I'd say the symptoms tripled in severity. Many nights, I'd spend between 2 to 6 hours in the bathroom. Some nights I slept in the bathroom to maybe save an accident or save time, I guess, because I didn't have much time sometimes when I woke up. I was still working, and it was getting very difficult for me. If I got to work and I got sick there, they only had one restroom, so I couldn't tie it up for an hour or more. So I'd try to get home, which was about 8 miles away. Most of the time I made it. Sometimes I didn't. I had to go wherever I went, whether it was in the forest or at McDonald's or wherever. I went through a lot of clothing. It says that, and at this point in my life, I felt like I was dying of the worst flu you could ever imagine. And I mean, that's exactly what I mean. It was horrible. Between 60 and 63, I was getting new symptoms, like polyneuropathy in my feet and legs. I was stumbling and taking some bad spills. I didn't feel comfortable even carrying my grandchildren. The neuropathy was causing more and more pain to the point that I had to take strong pain relievers, which brought on their own set of problems. Also getting leg cramps, like my mother and my uncles got. They were so severe that they dropped me right to the ground. You couldn't stand with them. And I could get them in 4 different places at once. I mean, I can't even tell you the pain. It was almost a deal killer for me. Sorry. I was hoping the amyloid would stay out of my heart no such luck. And those symptoms were causing me dizziness and exhaustion. As if that wasn't enough, I was also now suffering with a sexual dysfunction, which was very devastating. By this time, my life had come full circle. I was back in diapers, couldn't walk very well, and I'd lost my job of 44 years. I was also losing a lot of weight, which was the final symptom of everyone in the family that I had lost to hATTR, including my sister Ginger. At this time, I'd given up all hope. I had a family meeting. I told them all that I didn't want a liver transplant, because to me, if there was a chance of staying the same, I didn't want to stay the same. I did not want to go through that. And I told them, I said, I'm done. But shortly after that, a friend of mine and the founder of amyloidosis support groups, Muriel Finkel, insisted, she would not take no for an answer, that I go to a support meeting in California. So I went, and it was kind of a bad trip. Anyway, we made it there, got there late because I got sick, but anyway, I met Dr. Muriel Gertz there, and that's where I learned about the inotersen drug trial. He urged me to come to the Mayo Clinic, and he set up all the appointments to have the biopsies I needed to qualify for the trial. And some of those flights to Mayo were awful. I spent more time in the restroom than I did in my seats. True story. But after being on this drug for 4 years, I feel like maybe I'm more of a professional here on what you guys are asking than anybody. I'm doing better in all my symptoms, thanks to Axia's inotersen drug. My quality of life is much better, and I'm glad to be here now. I'm glad to help raise my grandchildren and watch them grow up. Since being on treatment, I've been able to regain back some control of my life. I've noticed improvements in my symptoms and the suffering, much less in the GI issues, which hasn't been brought up, and it was huge. I've gained weight back, which nobody in my family ever did once they went down on that track, and I'm now able to travel better, spend more time with my family and friends, without being afraid of having to spend half the time in a restroom. I'm very, very proud to be on this drug trial because my family needs it. Out of 24 family members tested, 18 are positive, including two of my 3 sons, my 2 other siblings, and now I fear for my grandchildren. As I discussed this with my son Bill one night, who was one that was tested positive, he said, we've lost so many family members already to this disease, and to actually have drugs that help gives our family great hope for the future. And he told me he was very proud of me for all that I went through. So lastly, I'd like to tell you that Muriel Finkel, Dr. Muriel Gertz, and Axia are my heroes. And my family, they saved my life. There's no doubt about it. And I hope to put ICER on that list too. I can't tell you how much has helped. I mean, when I went in there, I probably should have been on a cane to not fall, right? And I mean now, you know, and I can ride a motorcycle. Am I just better? I want to be here, guys. Honest to God, I would have done something about it. I would not have put my wife through what she went through with my mom and I, again. I would have handled it. I'd have probably done what my mom asked me to do. But anyway, I can't tell you the difference. I mean, I don't care what you put on that graph or who made it what. I mean, Lord God, this stuff saved my life. And I mean, my quality of life, I mean, from wanting to die, which nobody wants to die, right? I love my grandkids. So now, I want to live as long as I can. My grandson said, Daddy or Papa, I mean, will you be here until I'm 10? You know, he's like 6. I said, well, of course I'm going to be here. So I hope I am here.
Thank you.
Pray to you guys here. Thank you.
Thank you very much. Our next speaker will be Kristen Hsu. That was close. Thank you.
Good afternoon. My name is Kristen Hsu. I'm here on behalf of the Amyloidosis Research Consortium, or ARC. ARC was established as a nonprofit organization in 2015 to accelerate the development of and access to new and innovative treatments for systemic amyloidosis. We build collaborations across amyloidosis stakeholders to align research strategies, prioritizing the areas of research that will have the greatest impact on patients and improving patient outcomes. To do this, we focus on understanding patients' unmet needs and ensuring the successful approval, adoption, and diffusion of effective and therefore valuable new treatments test. We've conducted numerous studies and programs on patients' needs and preferences in relationship to treatment through patient panels, focus groups, and survey studies, and also hosted the the first externally led patient-focused drug development meeting with the FDA, producing the Voice of Patient report that was used to support the regulatory review of these products. Our mandate here today is to speak on behalf of patients and caregivers of patients living with hereditary TTR amyloidosis based on the data and stories we've gathered over the years. Now, in order to determine the value of any solution to a disease problem, it requires that we, one, understand the impact of the disease on patients and their families, and two, understand the solution's ability to provide outcomes that are meaningful to them. Hereditary TTR amyloidosis is an ultra-rare, progressive disease that is incurable and massively debilitating. It affects all domains of life and carries a high multi-systemic symptom burden, manifesting in neuropathy, fatigue, gastrointestinal events, fecal and urinary incontinence, erectile dysfunction, muscle weakness, pain, insomnia, fear, anxiety, depression, and vision problems, all of which are commonly cited as having an impact on daily functioning and quality of life in these patients. But I can't The impact of these symptoms is more than just physical. It's emotionally distressing, it's embarrassing, it's socially restrictive and work inhibitive. These symptoms have a cumulative and invasive impact on the patient's ability to lead a normal life. Patients have described feeling like a bystander within their own life, unable to actively participate. It also places a significant burden on the patient's family members as they provide physical and emotional care to patients while experiencing considerable emotional burden of their own. Family members often become full or part-time caregivers with consequences on their work, their social and financial situations. The hereditary nature of this disease families. Many patients have been caregivers for their loved ones. They've seen them waste away, and they know exactly what's in store for them. They live with the knowledge and dread of their own caregivers, seeing them succumb to the disease the way they saw their loved ones prior, and they live with the fear that they may pass the disease on to their children and future generations. There's a substantial lifelong impact on entire families. Now, patients have realistic expectations of these treatments. They know they're not cures, notwithstanding, but they can restore, prevent, and delay the deterioration of functioning, the benefits of which are considerable to both the individual patient and the population as a whole. Well, it's not for our ICER's methodology or determine what constitutes a reasonable price for these treatments. The cost-effective conclusions are a concern to us. We believe that ICER has taken a very cautious approach to the evidence of both treatments, particularly on the assessment of what constitutes clinically meaningful difference in these scales. We'd also emphasize that there are other hidden and unmeasurable benefits that aren't necessarily included in the overall clinical effectiveness rating, but have a huge impact on value for these patients. We urge everyone, the companies and payers, to work together to find a way to ensure that patients have access to these treatments, which are of high value to this patient group. We also can't overestimate the importance of choice and the respective value of each treatment. And while we understand that it wasn't the initial intent, it's disappointing that some of the debate and narrative surrounding the approval and assessment of these treatments has resulted in pitting one against the other. This clouds the importance of each treatment in its own right, and it's ultimately unhelpful to patients. We know that no single option for patients. Both treatments have considerable value, and the right outcome would be to find a solution that enables both treatments, which have numerous differences and very different value propositions to patients, not in the least where, how, and how often they're administered, to be available to physicians and their patients to make an informed choice about what is right for each individual.
Thank you very much. Our next presenter is Dawn Gimble Meyers.
I'll walk up here. Thank you.
Sure. Thanks, Dawn. Thank you for coming to Chicago.
Yeah, I have no conflict of interest. Is that ordering to just say it into the microphone?
No, that's it. It's up there.
Okay.
We're extra ready.
Extra careful. Thank you. I'm Dawn Gimble Meyers. I'm a 47-year-old hereditary amyloidosis patient. I am the first in my generation to benefit from an early diagnosis and early treatment. As a fifth-generation patient, I have experienced the cycle of caregiver to patient since I was 15. No one in my immediate family with hereditary amyloidosis has survived past the age of 56, even with a liver transplant. Each generation has experienced significant barriers in access to informed specialists, a proper diagnosis, health care, reimbursements, and a viable treatment. There are still so many barriers patients face before getting a diagnosis. Access to viable, life-saving treatment should not be the largest barrier to overcome. I stand before you today healthier than I was 1 year ago, and this has been reviewed by 3 specialists. I started on the Alnylam EAP program on August of 2017. Within 3 months, my NIS evaluation had dropped 8 points, and I too experienced significant drop in my symptoms. My daily life has improved significantly. As a stay-at-home parent, I no longer need a nap to get through the day. I complete 3 errands in a day rather than just 1. I no longer wake up in the middle of the night with neuromuscular pain. I no longer rise in the morning with clenched fists. I have regained feeling in my hands and my feet. We've gone on 4 different camping trips this summer as a family, and I hiked 7 miles. This was impossible last year. I have more energy to participate in my family life. Previous to treatment, I was becoming numb up to my wrists and my ankles, and our sweet family routine is near normal. Up until 2 months ago, I would fly out of state every 21 days so that I could get my treatment, and this was a cost we paid out of pocket. For our family, this was an investment that we treated as not an expense, investment in our future, an investment that included me in our future, that my husband would not be a widow, that my children would see real hope. My medical record would show disease regression, and it's important that we saw documented medical regression, disease regression, so that my payer would see this treatment as a financially better alternative than a liver transplant or any stabilizer currently on the market. Lack of reimbursement should not be a barrier to treatment or to my survival, nor put my family at financial risk. I have witnessed 3 generations go through the cycle of chronic illness, deterioration, and death. I currently have 2 cousins now who are 15 years post-transplant. They have lived long enough to have access to these new treatments. They are both now suffering from wild-type amyloidosis symptoms, and they will have access to new treatments, or will they die trying? I have been a hereditary amyloidosis patient since 2003 with my genopositive diagnosis. I have known what I didn't want, the emotional and financial chaos that I experienced as my mother's caregiver. I watched loved ones struggle with hereditary amyloidosis symptoms and healthcare insurance coverage. I am fearful of getting sick again, simply because the insurance market does not value or understand the benefits of my treatment. When my symptoms first presented, my then MO was perfectly happy giving me diflunisal, but incredibly inflexible and referring me out to the specialist I desperately needed to see. Will treatment be a matter of what's cheaper rather than what's effective? For my future, I wish to grow old with my husband. I want to see my children, Brant, who is age 10, and Genevieve, who is age 8, flourish and live their lives fully without the shadow of this disease. Access to an amyloidosis specialist for diagnosis can be the single most difficult journey a patient can have. For patients like myself, who have an extensive history, we know exactly when we need treatment, as soon as the hallmark symptoms present themselves. Without the benefit of the amyloid support group and relentless patient advocacy, many patients lose precious time due to a multitude of factors. The current cost structure for this treatment is too big to overcome if the insurance market does not value the treatment nor support reimbursement. Thank you.
Thank you, Dawn. Our last presenter is Dustin. Welcome.
I know I bucked protocol in doing this, but thank you for giving me the chance. You know, hope is a funny thing. One of my earliest memories of this disease was a family reunion in 1981. I was 5 or 6 years old, and like a lot of reunions, there's games, there's swimming, there's barbecue, except there was one noticeable difference. In the basement of the house we were at, there were 5 clinicians from Indiana University Medical Center. They're collecting blood samples from family members. They were collecting blood samples. In fact, my family has been doing this for decades prior, as our specific mutation of amyloidosis was first reported in 1956 as the Indiana-Swiss kindred of this disease. As I became older, I remember going to 2 funerals for my grandpa's brothers. One passed away at 54, the other one at 63. I can't tell you the first time I heard the word amyloid because I've just been around it my entire life. By the time my grandpa was in his mid-50s, it was obvious the disease was taking over his body. I have a love of golf because of my grandpa, because he used to take me out when I was real little and played. I remember the last time we played golf, it was about a year before he passed away. He would simply ride in the cart and we would hand him his putter and pull him up to the green so he could still putt. I remember being 17 and having to help my grandpa go to the bathroom in a bedpan. And in a moment that I know was embarrassing for him telling me how much he loved me. My grandpa died 3 months after I graduated high school at age 61. And while the polyneuropathy was devastating, his heart weighed heavy on him. Literally, it was 900 grams at the time of his death. In my early teens, it became just as obvious watching my dad that he had the disease as well. I remember him never sleeping well because his hands always hurt. 2 rounds of carpal tunnel surgery in his 30s and his 40s to try to alleviate the pain. The disease through my dad much quicker than my grandpa. I have no doubt that his hands and that his feet hurt, but he never had that experience. It was only in his hands because the disease progressed too fast for him to have those symptoms. At 53, 5 weeks after the birth of his 4th grandson, coming up on 9 years in November, my dad passed away from congestive heart failure, his heart weighing in at over 600 grams. Several years later, my uncle at 58 passed away at the Mayo Clinic in Florida waiting for a double transplant for a heart and liver. In 2003, I had 2 events that changed my life forever. Nearly back to back. On May 16th, my first son was born, and 2 weeks later, I received an email confirmation from Dr. Merrill Benson confirming the blood sample I sent him tested positive for the genetic mutation. In 2013, after my first carpal tunnel surgery, my amyloidosis was confirmed in a biopsy. As I said, I've been on this disease my whole life, as have all these other patients here today. While the details of each story is different, the themes are the same. Hard conversations, grieving what won't be in life, experiencing the debilitating outcomes of the disease, and dealing with death way too prematurely. That brings me back to hope, because today for me, hope looks like this meeting. As someone who's lived in around this disease my whole life, thank you. Thank you. Thank you for the doctors and the companies for their work to get us here. Thank you for the families who have been caregivers for this awful disease. Thanks to those who raised their hand and said, I'll take an unknown drug, not for my sake, for the benefit of my children and my grandchildren. Thanks to those who have given tissue and blood samples, and like my dad and grandfather, their bodies after their death for research. All of that has gotten us here today. Well, this meeting looks like hope to me. It's even more hopeful for our sons, ages 15, 13, 11, and 9, and future generations. Hope for a full, long life. Hope of not having to decide if or how many children you should have because of a diagnosis. While beauty is in the eye of the beholder, so is improvement. My standard isn't just my life. It's my life versus my dad's at my age. It's not just about me. It's about my boys, my future grandkids, and my great-grandkids. Any conversations and decisions today have to be made in light of this being a long-term game for the generations ahead. Thanks.
So now we're going to switch to some comments from our manufacturer partners. And if I can ask them to come up to the main session table here in the front. We have Sonali Agrawal and then Spencer Guthrie. Thank you so much for coming to Chicago today and being with us. Sonali, if you don't mind, will you go first? I say that?
Yeah,
Sonali. Remarks limited to about 5 minutes, if that's okay. Thank you.
Definitely. I mean,
Turn your mic on.
Hello? Okay. Thank you for this opportunity. Hearing the patient stories was incredible. I'm trying to control my emotions before talking when I'm supposed to talk. But I'm Sonali Agrawal. I'm Head of Value on Evidence Strategy at Alnylam Pharmaceuticals. Alnylam is a biopharmaceutical company committed to developing transformative medicines. We are also a flagship member of ICER and firmly believe in delivering value to patients and society. We thank both ICER for hosting today's discussion and also the patient and provider communities for sharing their insights and feedback during this process. As you've heard, hATTR amyloidosis is an extremely rare, rapidly progressing, multi-system disease that impacts all aspects of patients' life in a US population of about 3,000 diagnosed patients. Moreover, this hereditary disease impacts generations of families. Onpattro is the first and only product approved by the FDA for this devastating disease. To get has invested 16 years and more than $2.5 billion, translating a Nobel Prize-winning technology into a new class of therapies based on RNAi interference. Beyond Onpattro, Alnylam continues to develop innovative RNAi-based medicines to treat serious diseases at need. Until the recent approval of Onpattro, the management of this disease consisted of symptomatic treatments or off-label medical treatments. Steady disease progression robbed patients of the ability to perform the most basic daily activities. In contrast, Onpattro has demonstrated the ability to disease progression or reverse neuropathy impairment in majority of the patients. This offers patients the potential to maintain or regain their ability to walk, eat, bathe, and prolong independence, a dramatic shift from the current course of the disease. We commend ICER for thoroughly describing this highly complex disease and recognizing the clinical benefit of Onpattro. ICER also noted that the net price of Onpattro met affordability thresholds of the US health system. However, ICER recommends unrealistic price discounts between 90 to 95%, which do not align with either its clinical assessment or its rare disease framework. In other words, ICER believes that a breakthrough treatment with the ability to halt or reverse disease progression for a rare and often fatal disease and no other FDA-approved medical treatments should cost less than the average annual treatment for more widespread chronic conditions like or psoriasis. We question this conclusion when ICER has publicly acknowledged that treatments for ultra-rare diseases need a differentiated set of considerations. Additionally, ICER's framework inadequately captures broader societal benefits of Onpattro, such as maintaining independence, lost productivity, reducing burden, and improving emotional well-being, which are very important in this disease. Moreover, ICER's conclusions are based on $150,000 per QALY, the threshold used in standard value assessment. In previous reviews using its rare disease framework, ICER applied a threshold more than 3 times higher, which is $500,000 per. It is unclear why ICER would apply a framework specific to an ultra-rare condition and then recommend discounts based on a standard QALY threshold. Suggesting a 95% discount on a novel rare disease treatment is not a realistic solution to help patients gain access to life-changing treatments while maintaining innovation. Given the opportunity to transform care for people with hATTR amyloidosis, Alnylam is taking action to address barriers to access. We announced a patient access philosophy to help patients with comprehensive patient support services, engage insurers in open dialogue around value-based agreements, and pledge against arbitrary price increases. In fact, at approval, we announced several value-based agreements in principle for Onpattro. Insurers have vocalized our support for this approach, a fact that's not accounted in ICER's framework. When a breakthrough medicine changes the course of disease, we must ensure it reaches the people in need. By sharing our principles, Alnylam's focus remains on ensuring patients get access to these drugs and hope to inspire future value-based collaborations between insurers and developers. Thank you.
Thank you very much. Spencer?
Eric, I just want to respond to that.
I'm sorry, Dan.
A couple of comments there. So, note, because I'm sure the panel's read every single page of the report. Our threshold analyses in this case, as in all other cases with ultra-rare disease, did include cost-effectiveness thresholds higher than $150,000 up to $500,000 per QALY, so that information is available. We also applied standard language in the report, as we did with other reports, around how some decision makers do consider higher thresholds when making that decision. But our value-based framework, our value-based price range does not differ regardless of whether the condition is considered ultra-rare or not. It remains $100,000 to $150,000 per quality gained.
Thank you. Spencer?
Yes, thank you very much for inviting us as ICER to be here. My name is Spencer Guthrie. I'm a neuroscientist with public health training background. I have worked in clinical drug development for about 20 years. I'm here on behalf of ICER and to represent the value framework for inotersen. So I'd like to thank ICER for the opportunity to comment on these evidence report and to highlight to ICER and the public our company's mission to transform the treatment of serious and rare diseases with leading-edge RNA-targeted medicines. Patients are and always will be the focus of our efforts in developing these therapies, and it was, certainly amazing to witness the patient stories today. So it's really hard to talk after that as well. So I have a prepared statement, but I would also like to address some comments that came up this morning where I think there might be some inaccuracies. So one thing I want to address is PND staging and FAP staging is not an efficacy tool. It is a staging tool to be used at the beginning to stage patients to determine what outcomes might be. It is sometimes measured, as Dr. Burke mentioned, for historical purposes, but it's not used for this. It's an extremely blunt tool. There are staging, if you look at these two trials, for instance, there's a difference in the amount of patients in each stage. But if you look at the clinical, actually very similar. When trials are done in different regions, the staging can also be done differently in those regions. I'd like to point out another thing that came up this morning, and there was an inaccuracy that said patients on inotersen did worse than placebo on PND staging. That's incorrect. 10% improved their PND staging on inotersen versus 0% on placebo. So that's important for people to know. And that data is public. It's in the EMA documents. Also like to point out that 50% of patients improved on neuropathy quality of life, so through the Norfolk, and 50% of patients actually improved on SF36, which is a general health-related quality of life tool, which I'm sure you're all familiar with. I think there's wide debate on whether mNIS+7 is a validated tool. Within the amyloidosis community, it's certainly considered to be validated. Their minimally important difference of 2 points is considered to be validated. I would recommend a comment that was sent in from Dr. Peter Dick from Mayo, who is considered probably the world's leading peripheral neuropathy specialist. They worked on validating these scales. They trained all the centers to do the scales. They did central reviews of these scales, and they certainly consider, they certainly have a different opinion. As well, the model staging system that ICER has proposed to develop a new disease staging system. Using NT-proBNP, mixing staging systems between FAP and cardiac staging systems is not appropriate. It's not done by the experts, and NT-proBNP staging is specifically built for cardiac predominant patients. These are not cardiac predominant patients. Patients do have cardiac disease. This is a systemic disease, but there are very few patients in either of these studies and patients who have predominant polyneuropathy who have NT-proBNP greater than 3,000. So it's, from a modeling perspective, there's some problems with that. So we really appreciate being part of this, and I think anything that can bring awareness to amyloidosis, which I think you guys have done and we're doing today, is important because this is a underserved, under-recognized disease. But we do have some concerns about the report. I should say the statement, inotersen is an investigational drug. It's not approved, so the comments today are related to addressing ICER's evaluation. So specifically for that, it is under FDA review. So we think there's a few things. So there's premature assessment based on limited data set, an inappropriate comparison of efficacy between the two products, inappropriate classification of inotersen's clinical effectiveness, assessment of the risks associated with the therapies. There are significant assumptions underlying the methodology that need to be addressed, and we think that there's an incorrect use of 2 different best supportive care. So for disease, there's generally a best supportive care, but different supportive cares were used for this analysis. So even considering limitations of a small study, limitations of short treatment time frame, limitations of extreme heterogeneity between patients, we saw positive effects with inotersen across the spectrum of polyneuropathy clinical endpoints, neuropathy quality of life, general health-related quality of life. This was regardless of age, stage, mutation, prior ATTR medications, region, whether they had cardiac disease or not, and to see significance from such a small study just speaks to how robust the data set actually was. So with regards to comparison of the two drugs, appreciating that ICER has attempted to compare the two drugs, and in fact, they are compared. And so even this morning during the presentation, in the same statement, someone said, the patisiran may improve symptoms where inotersen doesn't. Those are two different scales. So you're measuring you're looking at two different endpoints and making an assessment and comparing two drugs, which is not methodologically sound. So the differences in these trials were very large. The regions that were represented, so 50% of patients in inotersen study were in the US, whereas 20% were in the patisiran study. The range of mutations was quite different in the two studies, and I think the time duration was different, the endpoints were different, so it's very important to understand this. And in terms of harms, while we agree that the harms that were spoken about with inotersen are fair, there was an enhanced monitoring program that was put in place for the second half of the inotersen trial, as well as the OLE study. The harms were much less, so we did not have the same side effect profile that was in the phase 3.
And then can we, maybe some of your other comments, we can move into a bit of a discussion. I know Dan may have some responses, or do you have any questions? We have a couple minutes here for a short panel discussion, but I think we need to move in.
Yeah, very brief response. So in terms of the P&D stage change information, I believe Karen's comment in the evidence presentation was around the percentages that worsened. Note that we recalculated the percentages from the EMA report because they did not include the portion of patients who had data missing in both trials, so those were recalculated. With regard to the point that you made about NT-proBNP, so we were trying to make the model reflective of what might be the experience in the US because the genetic mutations that give rise to cardiomyopathy are more common, but that being said, that the trial data were used, so that only patients who had that level of involvement in the trials were aspect. So again, we are very trial-based analysis. I know that the mind may stray, but every comparison in our presentation was for the active agent versus their comparator in that trial, not across agents.
Yeah.
Are there any comments from members of the CEPAC? Ryan, please, sorry.
I just had a quick question. Neither of you seem to have issue or bring up issue with ICER's annual drug cost, $345,000.
Our drug's not approved, and we don't have a price. So.
And we publicly announced that's our net price, which is $345,000.
Okay. And you did indicate that the research and development costs to go into it were $2.5 million?
$2.5 billion.
Billion or billion?
Billion.
Billion, thank you.
Any questions?
So with the comment about V122I, this was another, I'm sorry, with the prepared remarks, I could have gotten 5 minutes, but I had to address.
You did a great job, Spencer.
So just this one point is the most common mutation in the U.S. for patients who have cardiomyopathy is V30N. So we've done lots of surveys. So it's not, V122I is definitely the most common for cardiac predominant patients, but not for polyneuropathy predominant patients, which is where these drugs will be used.
And there's one thing to add to that. Isoleucine 122 is much more prevalent. 4% of the African American population has it, but the clinical expression of disease, penetrance, is unknown, but thought to be probably around 10 to 15% of those who have the mutation. So it may not be as big a cardiac population as initially thought.